So Georg is currently instructor at Johns Hopkins, but before getting there, he did his undergraduate degree in physics in 2010 from Heinrich Heine University in Düsseldorf, and then he did a graduate degree in natural sciences in 2015. After that, he followed up with 4 years as a postdoc with Richard Eden at Johns Hopkins, and looks like he liked it there, so he stayed on. And in 2020, he started with a faculty position in the Department of Radiology and Radiological Science. Now, Georg's research is in spectroscopy, which is not something that our lab does at the moment. But on the other hand, Georg has a big interest in standardizing the field, opening code, and this is where we have quite a lot of things in common. So I thought that it would be very useful to hear his vision for his field, for spectroscopy, but maybe for MRI in general. And then after the presentation, Georg will stick around, he'll answer questions, and anybody that would like to meet with him and discuss more is very welcome to stay on this channel. So without further ado, Georg, welcome, and we look forward to your presentation.
Thank you so much, Nicola. I think you should all be able to see the title slide. Is that correct?
Yep.
Excellent. Cool. Great. So yeah, thanks for the really nice and kind introduction, and thank you obviously to Nicola and to Julien for hosting, and to you all for attending. Would have been lovely to do this in person, but maybe we can save that for another day. So as Nicola said, well, we in MR spectroscopy, well, we are doing some sort of quantitative imaging as well, even if we're not doing imaging per se at all times. And I think that we face kind of similar struggles that the quantitative imaging community has faced and is still facing. We'll come towards that at the end of my talk. So it's my hope today, as Nicola said, that we can find some common ground for discussion, some similarities, and maybe even some inspiration. I can say that I've certainly been inspired by the work that came out of your group so far. I usually like to kind of start my talks with asking about who's vaguely more or less familiar with MRS. It's obviously a bit more difficult in this form, so it may be a little less interactive than usually. I hope that you will still take some things away about spectroscopy today, even if it's just peripherally touching on what you do. But if there's an urgent question, so please do unmute and shout at me. Ask a question in the chat. That's all fine. Usually, as I said, it's quite interactive and hopefully a little bit of give and take and push and pull. So what we're going to do today is I'll first give a brief elevator talk about MR spectroscopy, what it's about, why it's interesting, particularly the specific techniques of spectral editing, which has been my area of research for the past couple of years. I'm then going to present you with a few studies that exemplify the usefulness of these advanced techniques and applications. After that, I'm going to go into a little bit more detail about the methods developments that I've pursued during my postdoc years. And finally, as Nicola has already announced, I'll spend a few minutes about recent efforts to standardize the methods that we use, something that's a big challenge in all of neuroimaging and even more so in MRS, as we shall see. So a quick primer, most of what follows will concern clinical field strength. We do a little bit of 7T, but mostly we like to do our experiments at 3T. So obviously many of us use MRI on a daily basis, whether that be functional imaging or diffusion weighted imaging. And in almost all cases in proton imaging, most of these techniques are in one way or another based on measuring the signal that we receive from water, right? So if we plot the signal from the brain over the resonance frequency, which in spectroscopy we like to give in units of parts per million, we usually see something like this, like one strong signal from the water protons. But water is obviously not the only compound in the brain containing protons. It's the most abundant one, but there are many other compounds containing protons. So we can zoom into this seemingly empty area of the spectrum here. And when we do that, we will see something like this on the right-hand side. So this is a spectrum at 11.7 Tesla from the rat brain. And so we know that protons will produce signals at slightly different frequencies depending on the chemical environment that they're in. And we know fairly well in theory which of these peaks represent which compound, right? So we get characteristic spectral signatures for different molecules, and these different molecules that are visible to us in the proton spectrum are involved in multiple physiological processes. So there's molecules involved in neurotransmission, cell volume regulation, cell proliferation. Antioxidants are very important that scavenge free oxygen radicals, tumor mutation markers. So there's a plethora of information in there for us to mine. But the trouble is obviously that there is still overlap between these signals, and that overlap gets worse at lower field strengths, right? So this is 11.7 Tesla. And you'll see on the next slide how these things look at 3 Tesla. And this is a real challenge that underlies a lot of the methods development that we do, particularly at lower field strength. So this is what this typically looks like from a human at 3 Tesla. So we have some single strong peaks that still stand out, but there's this huge thicket of signals, and they're really, really hard to resolve from each other. They're small signals with lots of complicated peaks. So we need to somehow simplify these spectra and pull out the signals that we're interested in, for example, from GABA or from glutamate, everything that's going on with these heavily overlapping areas there. Just as an example, as I said, there's GABA, glutamate and glutamine, glutathione, the antioxidant, aspartate, ascorbate. Now, we can improve the separation of these overlapping signals with a technique that we call spectral editing. And at this point of the talk, I frequently go a bit deeper into the physics and the chemistry and the quantum mechanics, but I'll spare you these details today and instead just give an analogy. So what we do in spectral editing is conceptually not so different from arterial spin labeling, which you might be more familiar with. So in ASL, you record the same image twice, right? So once without doing anything, and then once with a labeling pulse that tags the incoming blood in the neck, right? And then you subtract the 2 images from one another, and the difference will only contain components that have been affected by the labeling pulse, and you'll get an image that is weighted by perfusion, right? We can do a similar thing in spectral edited MRS. We first acquire a spectrum where we don't do much, and then we record another one, but only this time with a selective pulse that we call editing pulse. And that pulse can be designed in a selective way so that it only targets certain molecules, but leaves others virtually untouched. And now upon subtracting these 2 spectra, we can remove strong signals like creatine, which is this strong signal here at 3 ppm, and we can reveal the underlying GABA signal that is here in green, right? So the shared concept is that if you want to resolve something small that's underneath a large background signal, you selectively change the small thing, but not the background, and then you subtract the background signal out, right? So that is one way to get at these tiny signals at field strength where they normally can't be resolved from one another. So one thing that is useful to know about MRS in general, as I said, it's not necessarily an imaging technique. There are ways to couple these kinds of acquisitions with spatial encoding, but the technique that is most widely used still and that we do most of our development in is called single-voxel MRS. So the way that the signal is being localized is essentially by 3 intersecting slabs. So first of all, we start with a slice selective 90-degree pulse here to excite a slice. Right, then we add another slice selective refocusing pulse a bit later with an orthogonal slice selection gradient, and that selects a slab at the intersection of the 2 slices. And after half the echo time, we add another slice selective refocusing pulse in the remaining slice selection direction, and that reduces our excited volume to a cube. So this is a typical volume that we acquire signal from, and for the low concentration metabolites that we like to look at, this is usually on the order of magnitude of 3 by 3 by 3 centimeters, or 2.5 by 2.5 by 2.5 centimeters. So the spatial resolution is usually quite coarse with these methods. In sequence diagram form, our spectral editing experiment will look like this. We first acquire, as I said, the normal spectrum, as previously seen, where we don't do anything special, we just localize it. And then we run the same experiment again, only this time we add our editing pulses. So we have 1 spectrum with the editing pulse on and 1 spectrum with the editing pulse off. We need 2 editing pulses interleaved between the localization pulses that we just saw on the previous slide. You can do this with a single editing pulse, but that introduces a frequency-dependent phase shift. So that is something that you have to correct for. So historically, this is the so-called famous megapress experiment, and this was just designed with 2 editing pulses, and this is still done like this to this day. As I mentioned before, when we subtract these 2 halves of the experiment from one another, we will arrive at the difference spectrum. And in this case, this is a difference spectrum that is spectral edited for GABA. And we can have a closer look at what makes up this spectrum. It has obviously only contributions from molecules with signals that were affected by the editing pulse that we irradiated, be it directly, like the negative blue signal from N-acetyl aspartate here that is directly hit by this editing pulse and is being inverted, or indirectly through coupling effects, for example, as the brown signal from glutamate here. There's also glutamine and glutathione, which is not included in this cartoon, but there are also signals from these. And it's worth noting at this point already, because we'll come back later to it, that the signal at 3 ppm, so this hump here, it still isn't pure GABA, despite the experiment being designed for it. It contains a broad underlying component that you see here in gray from co-edited macromolecules. So this is a serious problem that's plagued the whole field of MRS for a long time. There are these very poorly defined signals from macromolecules and proteins that contribute to the signal that you see, but they are very, very poorly understood and not well characterized. So they're really hard to disentangle from the actual metabolite signals that you want to detect. And that problem is so persistent that in this case, it is often just acknowledged as a limitation, and just the combination of these 2 compounds, so GABA plus macromolecules, is reported. And that's why in literature, you will frequently see something called GABA plus, which literally just means everything, all the signals that appear here in this 3 ppm hump. Let's just quickly talk about how much time we need to acquire a different spectrum, as we just saw. As I said, we're looking at signals with much, much lower concentration than water, about 50,000 times lower. So a single shot spectrum almost disappears in the noise, and we have to average a lot. For edited MRS, we obviously have to average even more because the signals are so tiny. We know that SNR is directly proportional to the volume of the voxel, but only proportional to the square root of the number of averages that we do. So if you want to gain spatial sensitivity, you pay an extremely high price by making your measurements so much longer. So these are typical parameters for acquisition. We have to average usually over about 300 times. And that results with a TR of 2 seconds and about 10 to 11 minute long scans. And as you saw earlier, these are quite large volumes, right? So this is pretty much the largest complication that we have to deal with. Spectral editing is highly specific to a particular molecule, but like the other MRS techniques, it's even exacerbated for spectral editing at the expense of temporal and spatial resolution. So we have a window onto these extreme low concentration compounds, but it comes at a cost. At this point, this is usually my first breakpoint, but it's just a quick recap. So MRS allows us to detect and quantify endogenous metabolites, usually at a detection limit of about 1 millimolar. Spectral editing unlocks additional metabolites that are otherwise almost impossible to detect because of their overlap with other signals. But all of this comes at a price at the extremely long measurement times. This is usually a good time to just pause for a bit, breathe, and ask whether there are any immediate questions emerging at this point.
So I'm curious, Georg, about the sequences that you're using.
Yes.
Looks like they've been around for a long time.
Yes.
How compatible are they across vendors? Do you have the same sequence on every site?
Yes, we'll come to that towards the end of the talk. I can say there are a few workhorse sequences that are somewhat standardized in terms of the localization and the sequence timing. They are about comparable, but there's still some subtle differences that you will notice and that might have an effect on the quantitative results of this. So pulses are different, sequence timings may slightly vary, gradient waveforms may slightly vary. All of that has an impact on the signal shape. We can try and account for that during modeling by simulating these conditions. it would still be desirable to have standardized sequences across vendors. It's something that the field is trying to take up. But the vendor native ones are still somewhat, as I say, they're largely similar, but different enough that you can notice it.
Security. There's a topic, food and security.
Can I follow up on that?
Absolutely.
Because that's a question we've obviously thought for all the quantitative MRI technique.
I think Eva is not
But
muted.
I mean, just having one topic.
No, it's on my.
Oh, it's
sorry.
sorry.
Oh,
Yeah, it's on my end. I'm sorry. The question is, yes, trying to standardize sequences, but given that the hardware differs, like the gradients, maximum gradient amplitude and other like hardware restrictions, is that a feasible goal?
Yes, I mean, you obviously have to settle for the kind of lowest common denominator. But the specifications are close enough, and gradient strength isn't really kind of the limitation factor for MRS experiments. Really, it's nice to have to crush signals that you don't want to have, but it's it's less important to max out the gradient strength than for diffusion interested folks, for example. So that is something that is achievable by taking a small hit maybe on some systems. I think that would be a feasible thing to do. Yeah. And these, as you will later see, these kind of standardized sequences exist already without massive hardware modifications that would be required. It's just a way of essentially, incentivizing people to use them and incentivizing vendors to build them into their systems permanently, other than just to distribute patches. But that's obviously not a concern that is singular to the MRS field. I'm sure you all are familiar with the same limitations, right? Cool. Any other immediate ones? Cool. So I hope that we all have some rough idea about what we do in MRS. And in this part, which I keep a little bit shorter because I wanted to focus more on the open source and standardization efforts that we as a field are trying to do. But still, let's have a little bit of a closer look at what we can do with this. So at this point, I'd like to share some applications work that is not necessarily mine, but some things that I find really exciting and that will just give you a good idea of what MRS and spectral editing in particular are capable of. So we've heard a lot about GABA in the example, and GABA was historically kind of the first application that the spectral editing technique was being used for. And it is still, I would say, for us as method developers, still kind of the number one thing that neuroscientists and clinical scientists ask us about. Well, we would like to measure GABA because it's involved in a lot of cognitive processing. So let's look at why that is the case. So GABA stands for gamma aminobutyric acid, and in the central nervous system, it is the most abundant inhibitory neurotransmitter. So obviously that means it takes part in the process of relaying information between neurons, as you all know. And the inhibitory bit of it means, well, it decreases neuronal activity by hyperpolarizing the membrane and making it less likely to reach the activation threshold potential. Now, an exemplary mechanism to really visualize this nicely is lateral inhibition. So any given stimulus will cause neuronal activity in the respective representation in cortex, so think visual or tactile or auditory stimulation. And lateral inhibition through lateral inhibitory interneurons will decrease the activity in the immediate neighborhood of the activated area, right? So we can look at a single, in this case, tactile stimulus as an example, and we immediately see the laterally inhibited area surrounding the activated area in neural representation space, right? So you can imagine now two stimuli activating neighboring areas in neural space, and GABA helps discerning these by suppressing the activation in the immediate environment of each stimulus, right? So if GABA and lateral inhibition are disrupted, our ability to differentiate between these stimuli will be impaired. So this is a very common thing that collaborators ask us about. Oops, that is not where I want it to be, is it? Well, no, I think we come later to this. Yes, so it's important to be careful in interpreting the signal that we get from measuring MRS, right? So the signal stems from all the GABA molecules in our selected region, but it is insensitive to whether they are within the synaptic terminals or in the synaptic cleft. We're not measuring active GABAergic neurotransmission. We're not measuring inhibitory activity per se. We're not measuring receptor density. We're measuring baseline GABA within this region. So we don't really have the spatial resolution, as I said earlier, that we would need to enable in order to be able to make inferences about lateral inhibition in particular. However, MRS levels have still been associated with the expression of the GAD1 gene, which is encoding glutamate decarboxylase. So this is one of the groups of enzymes that catalyze the production of GABA from glutamate. So it makes sense to have more GABA in our MRS measurements when this gene is expressed stronger. So generally, we like to use the term GABAergic tone to describe what we arrive at with our spectroscopic measurements. So it reflects the potential to perform inhibitory activity. And this is why, even though the GABA spectroscopy measure does not in itself reflect GABAergic neurotransmission or lateral inhibition, however, it still has functional and behavioral impact via, by being active and being actively involved in lateral inhibition, as we shall see in the next part. So a nice example for application of GABA spectroscopy is this study from my mentor and supervisor, Richard Eden, which he did a while ago. So they wanted to know whether GABA levels in the visual area have an impact on a specific visual orientation discrimination task. So this task is essentially that you as a subject are being presented two black and white gratings, and they are slightly rotated against each other, right? So by pressing a button, you're supposed to indicate whether you perceive this rotation as clockwise or counterclockwise. So it's essentially a measure of the resolution of your visual system. And it's done with a staircase design to determine kind of an individual rotation threshold where you as a subject can reliably and correctly recognize whether rotation in a certain direction has taken place. And during the study, also GABA spectroscopy was performed in the occipital part of the brain, as indicated here by this green box. And the result was quite fascinating. So the individual GABA levels predicted the individual orientation discrimination threshold here with better results, which means a lower threshold, that means higher resolution and perception of rotation, was associated with higher GABA values, right? So higher GABA values allowed you to perform this visual rotation discrimination task with greater accuracy and greater consistency. Now, to switch domains, the frequencies of stimuli are similarly encoded through GABAergic mechanism, so to say, over time. For example, tactile processing. So first, this is something that we've seen earlier. So spatial stimulus discrimination, or the discrimination of the stimulus representation in neural space, worsens when GABAergic neurotransmission is disrupted in an animal model. And what also happens is that the encoding of frequency information in neuronal activity gets lost. So these are, I believe, just, I don't know whether they're single cell recordings, but I'd have to look that up. But it seems that GABA in general is a critical ingredient to encoding frequency in neuronal patterns. And one can hypothesize, obviously, that GABA levels will influence an individual's ability to discern different frequencies of stimulation. So in this case, in this particular study that we're going to see, this is tactile stimulation, very similar to the orientation discrimination paradigm that we saw before. So how do we probe individual ability to discriminate tactile stimulation? Whether there are devices that are essentially vibrating stimulators on the finger. And again, the subject is asked to provide a response on, let's say, which of two closely following stimuli are perceived as having the higher frequency. So this study, performed by former colleague Nicholas Putz, who's now at UCL in London, it's fascinating. So this task was performed in right-handed participants. They also received GABA spectroscopy measures in the left sensory motor cortex and the occipital cortex as a control volume. And the finding is quite consistent with the visual paradigm, right? So more GABA in the sensory motor area corresponded to a lower frequency discrimination threshold. And that again means better performance in discriminating stimuli that have similar frequencies, right? And more astonishingly, there was no correlation with visual GABA. And that led to the conclusion that there is a region-specific functional impact of GABA levels. So there appears to be no uniform tonic inhibition level that is kind of valid throughout the brain, but it is rather region and task specific. And finally, so there's an example of how these kinds of experiments can also bridge into clinical research and help investigate pathophysiological mechanisms. So this is a study that I conducted during my PhD back then in Germany about patients with hepatic encephalopathy. So we had a large cohort of patients with liver cirrhosis, and hepatic encephalopathy is the major neurological complication of cirrhosis. So the liver loses its ability to filter toxic substances, and among others, ammonia is the one that does the most damage in the brain. So it's a very tiny molecule, can easily pass the blood-brain barrier, and it interacts with almost every system in the brain that you can imagine. So genetic expressions, enzyme production, neurotransmission, anything you name it, ammonia messes with it. So symptoms are usually cognitive and motor dysfunction of varying severity. There is minimal or subclinical HE, where the patients appear normal in everyday life, but they show impairments in psychometric testing. And then there are the grades between HE0 and HE4, starting with mild memory problems, trouble to stay focused, Parkinsonian, tremor, up to coma hepaticus. So it's a really, really drastic and devastating disease. However, there's a quick routine clinical test that is frequently used to get a first idea of the severity of HE, and that is the so-called critical flicker frequency test. So this is, interestingly, again, a frequency discrimination task, a bit like the two that we've seen before. So here you put on a mask like this, and you see a red dot, and this red dot oscillates at about 60 hertz. So you perceive it as a steady light. Right here, it's obviously a bit exacerbated for demonstrative purposes. Now, the frequency will decrease slowly, and you are instructed to press a button as soon as you start to perceive the light as a flicker, as right now. So this is obviously a measure of the temporal resolution of your visual system. And if you're healthy, you'll be able to discriminate higher frequencies. And this is precisely what we see. So this critical flicker frequency threshold decreases with increasing severity of the disease. So that means that the temporal resolution of the visual area in liver patients worsens. And now, given that we've seen that GABA seems to be able to predict the performance in tasks like these, what we did back then was we wanted to have a closer look and did exactly the same thing as in the previous studies. So we performed this flicker frequency test as well as GABA spectroscopy in a number of patients and controls. We also sampled their blood because we wanted to know, well, how high are the ammonia levels? How big is the degree of liver damage in this case? And the results aren't even so surprising anymore. So even the patients without everyday impairments showed already markedly decreased occipital GABA levels. As we hypothesized, the occipital GABA levels did predict this critical flicker frequency, and again with more GABA predicting better frequency discrimination performance. So GABA in this case again was kind of a to be required in order to be able to perform well. And finally, GABA was directly correlated with the ammonia load in the blood, so indicating that the degree of liver damage has a direct impact on brain GABA levels, which is kind of exciting and also a good example for what is becoming more and more interesting to researchers as an example of the so-called brain-gut axis. Finally, I thought a nice bridge over to the next part of the talk would be this example of what I see as the potential killer application to make into the clinics. In brain tumors, there are two mutations that are associated with improved survival. So that's the so-called IDH mutation and the 1p19q codeletion. And so much so that they are now part of the WHO classification scheme. As you can see here, the median survival of glioma with wild-type IDH is about 15 months. That is the yellow plot. Now, the IDH mutation makes tumors more susceptible to radio and chemotherapy. and median survival increases to 80 months. And finally, if the 1p19q co-deletion comes on top of that, survival increases to 134 months. So there's 10 times more than the wild type. And this is obviously something that you would want to know about a brain tumor. Does it have these mutations? It also affects the treatment regimen and the prognosis, clearly. Now, the cool thing is that without changing megapress for GABA acquisition parameters, you can find 2 chemical substances that are indicative of these mutations. So it's a really fortunate coincidence owed to the fact that both of these chemicals have similarities to the GABA and the glutathione molecules. So first, there is 2-hydroxyglutarate, which is this tiny peak here, around 4 ppm. And this shows up only if a mutation on the IDH enzyme is present, but it doesn't show at all in healthy tissue or in tumors that don't have this IDH mutation. Cystathionine, so this one here in green, is produced if the tumor shows the 1p19q co-deletion, and the signal shows up at 2.6 ppm. Again, you don't even need to change anything. You can just take your stock GABA detecting sequence and run this in tumor, and you have a technique to determine tumor mutation status. So I argue this is one of the things that needs to be brought to clinical application as soon as possible. And for that, as we will get to see later, what we will really need is kind of a standardized acquisition and analysis protocol. So just this as a brief excursion into the potential applications. We've seen that particularly MRSHub GABA can provide a link between what happens on a molecular micro level and at a behavioral level. So it's kind of a nice translational bridging techniques. But it also, apart from GABA, there's multiple molecules that are, as we said earlier, involved in very specific processes and are being actively investigated in research in neurodegenerative disorders, neurodevelopmental disorders, psychiatric disorders. And a lot of these molecules are only really accessible to us if we manage to do spectral editing experiments on them. All right, this is another opportunity for a quick break and any questions that may emerge at this point. Cool. Okay. Then, Nicola, did you have a question?
We're good. We're good.
Okay, cool. Then we'll just move on, and I will keep this chapter a little bit shorter than usually, because I think that the last one that I've prepared is a little bit more exciting for our getting together. So as I said, we just talked about GABA a lot, and it remains the most frequently measured compound with spectral editing, but we've also seen it's not very time efficient. So the centerpiece of my postdoc work was essentially to answer the question, how do we get more efficient with these types of experiments? As I said, MRS can't be inherently accelerated. We're just dealing with a very low signal to noise, and these experiments are highly tailored to a particular molecule of interest. And interestingly, the way that this megapress experiment for GABA was being done hasn't changed dramatically since it was first done in 1993. So essentially, the question that we've always been trying to answer, if we have to spend 10 minutes on an acquisition to get GABA, well, can we be more efficient, or can we be less wasteful with our time? Can we extract more information from this 10-minute measurement that we have to do anyway if we want to get one measurement of GABA? The first question that we asked and answered was, can we do this whole thing in more than one region at the same time? And the answer was yes. We make use of a concept that borrows from parallel imaging. Essentially, we're using dual band excitation pulses to generate signal not only in 1 but in 2 voxels at the same time. And we're using spatial coil sensitivity profiles of the head coil to determine where the received signals come from. And we can use this reconstruction to disentangle the signals and correctly assign them to where they come from. So this works quite nicely. We're getting nice spectra at full signal to noise ratio, provided there is enough separation of these 2 voxels so that the G factor is favorable. The kicker is we get both of this at the same time. So we're getting 2 measurements worth of data in the time that it takes to do 1 measurement, right? So we've demonstrated this is compatible with spectral editing, and we already gain a 2-fold increase in data acquisition rate. So that's kind of the endeavor. How do we make editing more efficient in the spatial domain? There's also a metabolic domain to this whole thing. I mentioned earlier that many molecules are low concentration and require spectral editing. So beyond GABA, For example, we have glutathione, the antioxidant, there's lactate, the anaerobic product, NAAG, a modulator of glutamatergic neurotransmission, 2-HG, which you just saw. So I don't want to bore you with the details too much, but as you can see from the cartoons, all of these have different chemical structures. And megapress for GABA won't work optimally for all of them, even though it happens to accidentally work for 2-HG. But normally there is a set of editing parameters that is optimized for one metabolite only, but not for others, which is why we have to do these extremely tailored experiments in the first place. So in an attempt to battle this, we took a step back and essentially said, well, we have to perform these experiments in this alternating fashion of 300 averages anyway. So these are the first 4 averages of a GABA-edited megapress acquisition. So we switch on the editing pulse, then we switch it off in the next average, we switch it on and we switch it off, we do the subtraction, and we end up with a GABA-edited spectrum. Now, if we wanted to edit for glutathione or GSH, the signal that we want to observe is also at 3 ppm. But in order to isolate this signal, we need to apply this editing pulse at a different frequency, in this case at 4.56 ppm, right? So we do essentially the same thing, whether we want to edit for GABA or for GSH, but we do the intervention, so to say, on opposite ends of the spectrum. And now the question was, can we actually maybe do this simultaneously in one single experiment? So we actually really did merge these 2 experiments that are happening at the opposite ends of the frequency spectrum into one big experiment. And all that we really needed was a dual band editing pulse, which essentially works both at 4.56 ppm and at 1.9 ppm to change the metabolic signatures of GABA and of glutathione. So now essentially we carry out 2 megapress experiments at the same time. We get full signal to noise for each of these kind of sub-acquisitions. But instead of a 2-step experiment, we have a 4-step experiment. And just by cleverly reconstructing the 4 steps of these experiments, we can arrive at the same GABA-edited difference spectrum that we see here on the bottom. That is exactly the same as if we would only do GABA megapress. And with a different reconstruction, we arrive at a glutathione-edited spectrum that we can see here at the bottom. And again, the kicker of all of this is that we essentially perform 2 experiments at the same time, but in the time that it takes usually to perform one of these experiments. So it's again a 2-fold increase in data acquisition rate. So it's quite cool. It's a substantial increase of data acquisition rate. It's already useful, but we took a step back and we compiled a list of all the metabolites that you can look at with spectral editing. And we designed the ultimate experiment, essentially. It can detect GABA, it can detect glutathione, it can detect ascorbate or vitamin C, it can detect N-acetyl-aspartyl glutamate, it can detect aspartate, it can detect lactate, 2-hydroxyglutarate, and cystathionine. So we can measure all these metabolites within a single experiment just by cleverly placing additional editing pulses throughout the acquisition, and then by cleverly combining the resulting spectra that we get from each of these sub-acquisitions. This is really as far as we can take the whole thing. So we have a pulse at 1.9 ppm, one at 4.56, and another one at 4.16. And all that we really have to do is to take these 4 subspectra that we will get and combine them in clever reasons that allow us to put the edited contributions from each signal into different reconstructions. So you'll see that the GABA and the 2-HG signals, they end up very prominently here in this reconstruction in the center, whereas the signal from glutathione, which also happens to be at 3 ppm, appears over here. Whoops, am I right? Appears over here at 3 ppm in a different reconstruction, as does lactate, as does N-acetyl-aspartyl glutamate. So we essentially solve the problem of overlapping chemical signatures by selectively manipulating them throughout the experiment and separating them out into different parts of the reconstruction so that we can then model and quantify them separately without the overlap. So that is something that affords us essentially a 7 Tesla-like resolution of chemicals that are otherwise really difficult to tell apart. We still get everything for free that we get in conventional normal spectroscopy. So the strong signals from creatine, from choline, from myo-inositol. So it really supercharges the kinds of experiments that we can run and the amounts of hypotheses that we can test within a single acquisition. It's a really neat thing to do. All of this is then, again, works at 3 Tesla. And we have sequences essentially for any vendor ecosystem. So we're trying to distribute this equally and evenly among vendors. The final thing about this is we don't have to stop there. We can combine the spatial acceleration that we get with the metabolic acceleration that we get, because they essentially happen in orthogonal domains. One happens within one single TR, and the other one happens between two TRs. So we can achieve almost, well, in this case for Hermes, PRIAM is the combination that we've called it a 4-fold acceleration, but combining this with HERCULES affords us up to, I think, 14 or 16-fold acceleration of data acquisition rate. So it just changes the way that we think of these kinds of experiments. Instead of being targeted towards one signal metabolite, we much more think of it now as collecting a huge number of metabolites instead. So just a quick recap. Obviously, edited spectroscopy requires long measurement times. And if we have to measure for a long time, we might as well try and use them more efficiently to acquire data from more than one region at the same time, but also by encoding multiple simultaneous editing experiments with these new Hermes and HERCULES approaches. Cool. All right, this is the opportunity for another quick break and any questions that may have come up. And for me to take a sip.
How much of a pulse sequencing background, pulse sequence programming background did you have before doing this? Is this something that you also did during your?
Yeah, I had never done any sequence development before I came here, and so the way that these experiments were designed, it really only requires a few lines of code of kind of like adding these dual band pulses, which is essentially a cosine modulation in the time domain. So it's all doable. It doesn't require massive interventions on the sequence development side. I took the Philips pulse programming course and the sequence development course, and that was about it.
I have a quick question, like a follow-up question, actually, because
Sure.
I was not aware of this PRIAM technique, which seems really, really cool. Is that like analogous to the like parallel imaging, where you would basically use like different coil sensitivity profile to disentangle the contribution?
It's the same idea of multiband and parallel reconstruction. It's dense, yeah.
Right, so that to me is more involved than only a few lines of codes for doing those kind of experiments, right?
The problem there is the reconstruction. So for the acquisition, all that you really need to do was split the excitation pulse. Everything else stays the same. The problem is really then the reconstruction, and that is indeed a little bit more involved than a couple lines of code. Yes, yeah, that gave me a headache.
Right. No, this is a brilliant idea.
Yeah, it's well, we could talk about the shortcomings of this for quite a while. There are many of them. Primarily, for example, that you can't arbitrarily place these two voxels, so they are kind of co-localized because you're just splitting the excitation pulse, but the refocusing pulses stay still the same. So you always have them coplanar. So if you rotate one, the other moves. So it works well if you want to do kind of things that are symmetric between both hemispheres. If you want to say like left and right insular, for example, that works well. It's less useful for arbitrary voxel locations. Yeah. Cool. All right. Let's move on. This last bit, I think I'm already quite close to maxing out my time. So, but I thought this one was the one that's probably most likely to lead us into the discussion. So, you're all super convinced now, obviously, since this great talk, that MRS is a cool technique and we should all do it more often. But as you also know, well, or you may have asked yourself, well, why is nobody really doing this? Why don't I know anyone who's doing MRS? Interestingly, MRS was the first application of the NMR effect, so that came way before imaging, way before fMRI, way before DTI. So why did it fall so far behind in being widely used? And my main issue personally with MRS, despite working in it now for a decade, it is still not a replicable, a reproducible, and a comparable technique. Right? There are many underlying reasons, but ultimately what it means to radiologists, to clinicians, to researchers, there's just low clinical decision-making value in doing an MRS experiment. And the impact on clinical research is frankly also quite low, because it's just really difficult to run a study on MRS and collecting ample amounts of data, analyzing them. You always need to have a spectroscopist on the team to do all that for you. And, you know, acquisition-wise, there's just a lot of different techniques that are being used. They're all vendor and site-specific frequently. So you really need, as I said, you always need a spectroscopist to run a spectroscopy study. That's not how I would think of something that wants to be useful. It's even worse on the analysis side. So there are loads and loads and loads of software solutions out there, and essentially people do their own thing with it. There's no standardized processing pipeline, really. There's no agreed upon optimal way of doing it. There's no code and data sharing culture, really, so nobody knows what the other person does. And in the end, there's always, you know, numbers being pushed out there, but they just don't agree well with each other because, well, people use different ways to arrive at these numbers. Another problem, that the de facto analysis software that was considered the gold standard used to be a black box, which we'll come to in quite a bit. So there was never any scrutiny about whether the numbers that come out of these modeling algorithms are actually meaningful. And finally, with MRS, what's really, really difficult, it's almost impossible to validate, right? You're getting in vivo, well, you're getting numbers of concentrations of metabolites, but you have no way of knowing whether they're true. And none of these analysis methods have ever been truly tested, benchmarked, and validated. And all of this just leads to everything that's down here in the red circle. Nothing is really replicable or reproducible or comparable. Fortunately, the field is beginning to recognize this. And this is where, well, I like to see kind of a silver lining on the horizon. There's at least, I think it's like 11 or 12 consensus papers on various aspects of spectroscopy been formulated by experts who sat down together and said, well, we have to agree on something because otherwise this is just becoming irrelevant soon in the future, right? And so about acquisition and about analysis and about interpretation, about reporting of MRS experiments and results, there is some progress being made. For example, this is what we've been already kind of alluding to, something that emerged from the clinical consensus or from kind of the clinical perspective is, well, what we definitely need is some sort of standardized workhorse default acquisition that is somewhat the same between vendors. And so led by a group in Minnesota, together with a few people from us here at Hopkins, there is a standardized semi-laser sequence now that is readily available, and it virtually gives the same spectra from Phantom, whether you do it on a GE system, on a Philips, or on a Siemens system. So this is as harmonized as it can get in terms of pulses. Pulses are open source, so they're shareable. So this is as close as we can possibly get to a standardized acquisition. We have done a similar thing in the spectral editing realm, where we have teamed up with a bunch of industrial partners at the various vendors to create the standardized editing sequence on the right-hand side here. I'm not going to go into the detail about that. So that is acquisition. So we're moving slowly. Now it's all about getting uptake and getting people to actually use the standardized software. The next big problem is processing and modeling. So the way that a spectrum in spectroscopy is being modeled is usually by something that we call linear combination modeling. And the way this works is we approximate the spectrum that we have with a nonlinear least squares model. And essentially we let the model figure out how it can put together a measured spectrum as a linear combination of simulated basis functions for each metabolite that we think is in there. We can fortunately accurately simulate how an ideal response from a metabolite to a given pulse sequence looks like. And then the model usually accounts for, let's say, line broadening due to local field homogeneity, small frequency shifts, so on. So it all sounds relatively easy in theory, but it's really hard to do because it turns out there's a lot of things that make this process incredibly difficult. So most notably, there are these signals from macromolecules that I was talking about earlier in the beginning. They are really, really not well defined. There's rolling background changes that we need to account for with a smooth baseline. So mathematically, there are many, many possible solutions to any of this. And it's a really, really difficult, ill-posed, ill-defined optimization problem. And now we know all that, and yet still it's kind of the best way that we have. So we know that we have to employ a really complex modeling process to accurately model our data. Expert consensus recommends that we do this. Modeling is unfortunately only one part where all these uncertainties can pile up, because pre-processing and all of that, it also is highly unstandardized, as I just told you before, right? So everybody's essentially taking their raw data and processing them in the way that they want without really reporting how this is done. The most widely used software implementing the algorithm to solve the modeling problem was a tool called LC-Model. It's a software that's almost 30 years old, but still kind of the reference and the benchmark in quantification, until about 2 months ago, when the single developer of the de facto gold standard retired. So now we have a free tool with source code that is available, but there's no active development that is happening anymore. So that's a problem. Finally, the process is not really well understood, in part because it's such a complicated one. So we don't really know how do starting values and algorithmic decision-making impact the metabolite estimates that we get out of the modeling process. So all of this is just, it's really variable and up in the air. So as I said, there's multiple softwares who can do the task. It used to be LC-Model, which came at a price tag of $13,000. It is now open source, but it won't be continued anymore. So we now have a situation where the gold standard is discontinued, and we somehow need to find a way forward. Now, fortunately, over the last 2 years, we've seen several new toolboxes released, and they're all quite similar in conception and design. They're all running in different languages, which is quite awesome. So I believe that we have a new wave, kind of a new generation of MRS data analysis ahead of us, all with the core premise that all of these softwares are transparent, open source, and reproducible. So our very own software, I'm just going to quickly rush through this, is the Osprey software, which we've developed with all of these premises in mind. It essentially automates the entire workflow in a reproducible way by defining a job file, by loading the data from the raw file formats, by processing them according to current expert consensus, doing the linear combination modeling in a transparent way and modifiable way so that we can really see everything that's happened, other than an LC model where all of this was behind the closed source curtain. It interacts immediately with SPM functions, so you can co-register MRS voxels to anatomical images, do segmentations, pull in gray matter, white matter, and CSF fractions, and finally do quantification based on all of these metrics. So this just really quick, it has a graphical user interface that allows you to do a quality assessment. It interfaces directly, as I say, with any sort of nifty image that comes. It does an inherent image intensity analysis on any external image that you feed it. Well, we've benchmarked it against LC-Model and Tarquin, which is another popular open source algorithm to model data. Interestingly, or well, not surprisingly to experts, what we ultimately found is that there is some agreement in the estimations of strong signals such as N-acetyl aspartate, where the overlap in distributions is quite notable. Whereas as soon as you move over to more complex signals from myoinositol, for example, for glutamate and glutamine, the agreement between different tools on the exact same data that you give them is surprisingly low. So you introduce substantial variance just by choosing a different analysis tool, which is a problematic thing for a field, obviously. I'm going to skip this since we're running over. already a little bit. So just to close it, what we can do about this problematic state of affairs right now at a community level is found a committee that tries to tackle this and join forces. So we've founded an initiative coming out of the study group for spectroscopy in Montreal at ISMRM, fortunately. We've developed a plan to create resources that enable people to share their code and share their data. And we've been approved by the study group executive board, so we're a permanent standing committee. Interestingly, out of the 6 current members of it, 3, so myself and Will and Martin, are developers of this new wave of analysis software. So Will is the one developing FSL MRS, Martin Wilson is the one who's developed the SPAND package, the A-B-fit algorithm. We all have good working relationship with each other. We like to share stuff with each other. It makes me hopeful that together we can somehow create a better environment of methods and tools that will help the field address its shortcomings. So what we have created with the MRSHub is a common resource where people can share their data. We have a forum where they can discuss and exchange ideas and support, and finally a repository for data sharing. It's all on GitHub, it's all online, and it's been taken up by the community, well, better than I would have ever hoped for. There's clearly a need, especially by trainees that try to orient themselves in the field. That has been really a pleasure to see them accepting this and trying to get involved in the community and expressing their interest to contributing to shared resources and shared tools. Initiatives that came out of this include the nifty MRS specification, which we've been working on, trying to generate a standardized file format for MRS data, which suffers from the fact that every vendor has devised their own little text-based format how data is being stored. It makes exchanging data a real pain, and we have decided to augment the nifty specification, write a header file specification for that, and use it to store raw and processed spectroscopy data, and that works really nicely. We're collecting data sets that other people would otherwise not have access to, that are hard to acquire, and just give them ways to benchmark and test their own methods developments. And finally, kind of the vision that I would have for this field is to really cover all the bases. At some point to arrive at a completely vendor agnostic acquisition with common sequences, to have, well, large data sets of in vivo data, both from healthy volunteers across the lifespan, but also from pathologies. Consensus analysis methods that are portable and available across different software environments, maybe even in the cloud. Easy development of new analysis methods that can make use of large amounts of synthetic data that we can generate at the click of a button. We can train networks to do the analysis for us. We can benchmark our analysis methods. And finally, feed all of that back into a clinical workflow so that you don't need a spectroscopist anymore to really run and analyze a spectroscopy study. So that is my well, ambitious hope for the future, that we can arrive at something that looks like this. And I believe that this is probably something that you all have thought about in how can you make QMR techniques relevant and usable and user friendly, and how can you improve the uptake in the field. And well, it's partly why I'm hoping that we can all stay in touch for a long time in the future, because I believe that by joining forces we can have a great impact in our own little niches, but also beyond. Okay, so if you don't take anything else away, take away that MRS desperately needs to do something about its reproducibility crisis. One of the problems is that it's enormously difficult to acquire and model, and we need to be clear what we do and why we do it. But I'm hopeful that we're at the beginning of a new era and a new decade, hopefully, of these issues being addressed and of new robust methods being developed. And with that, I will acknowledge all my friends, lab partners, colleagues, and collaborators, particularly my postdoc, Helge Zollner. And I thank all of you for taking the time and going over the allotted time with me together. I apologize for that. I think it was 45 minutes. I'm very sorry. Yeah, if you have any more questions, I'm happy to take them now or whenever we have our individual chats. And It was a great pleasure to be with you today. Thank you so much.